What's good, guys? It's me, Moe. A lot of you were wondering about if they can call me or text me for the help for the quiz, and I definitely can. No worries about that. But some won't be able to contact them, so I made this video real quick just to explain everything that we need to know. Right here with me, we have the DNA model. Okay, I'm gonna first start with labeling everything that there is. Okay, so I'm gonna get a black pen. I'm gonna lower this. We're gonna start labeling everything. Okay, this pentagon shape right here. There's a few of them found in the thing, right? I'm gonna just highlight these so you guys can see. So we get a sugar here. We get a sugar here, sugar here, sugar here, sugar here, sugar here, sugar here, and sugar here. Now, why is that important to know? What are these all highlighted for? Now, these are the sugars found. This is a sugar found in DNA. Now, it's a sugar because it's a carbohydrate. But this, you don't need to know that's a carbohydrate or it's a sugar. Well, it's technically, you just need to know it's a sugar. This pentagon itself, right, is called deoxyribose. That's what this sugar is called. Now, deoxyribose is found only in DNA. It has 5 carbons, so you can call it a 5-carbon sugar. And how do I know this? Now, you see every point here? Call that a C, a C, a C, a C, and C. Count how much C's are there. 1, 2, 3, 4, 5. It's a 5-carbon sugar. Just look at the shape and you can easily determine. For example, glucose is a hexagon shape, so you would call it a 6-carbon sugar. Okay? It's a 5-carbon sugar. In RNA, it's called ribose. Okay, know the difference. I'm gonna highlight this. Alright? In RNA, it's called ribose, and in DNA, it's called deoxyribose. That's why the actual name of DNA stands for deoxyribose nucleic acid, and the actual name for RNA is ribose nucleic acid. It's its major difference in the structure itself. Alright? Now that we got that concept cleared, we're now going to look into this yellow portion. Now, this portion isn't as important. All you need to know is this is considered as a phosphate group. So I'm going to write that down. Phosphate group. Okay? This is considered as a phosphate group. Like that. That's just considered as a phosphate group, and you don't need to know its function or anything about it during the DNA, during the actual quiz. You just need to know that connected to the sugar on the is a phosphate group, any nitrogenous base. Now we're going to get into the important part. But before we do, I want you to understand the type of bonds found in DNA, because this is also going to be on the test. The bonds found in this purple section between the bases are hydrogen bonds. We'll get into more detail on why they're so important later. But the bonds found in this section area, where it's connected to where the sugar, phosphate, and the nitrogenous bases are all connected here, this kind of shape, this lumpy shape, they're all connected by covalent bonds. So what I'm going to type right here is connected by covalent bonds. Right? And then this is hydrogen bonds. Now, let's go into the actual nitrogen bases, because this is basically going to cause everything that we know about DNA. But before we get that, let's just say one last thing. The structure shape of DNA is called a double helix. And it goes from a 5 to 3. And on the other strand, it will go 3 to 5. Now, you don't even personally know why it's a 5 to 3 and 3 to 5 until the actual DNA replication test. Okay, and when that happens, I'll go into a whole lot more detail on what that is. It's just something based on the sugar, where the carbon is in the strand. Don't need to memorize it. Don't need to know it. You just need to know that one strand goes 5 to 3, and the strand goes to 3 to 5. And now, because of that, it looks as if one strand is going this way, right? Because 5 to 3, while another strand is going to 3 to 5, right? this is technically parallel, but it's not purely parallel. So that's why we also call DNA an anti-parallel structure. So we also call it a anti-parallel structure, right? Now, the next thing you need to know, the actual nitrogenous bases. Now, what causes this double helix? It's actually the nitrogenous bases itself. Now, before we do that, remember that T, which stands for thymine, always connects to adenine, and it is only found in DNA. Thymine is always found in DNA. Only found in DNA, right? If it's RNA, if the structure is RNA, if the question asks, for example, in an RNA structure, adenine pairs with what? You would write uracil. Someone write that down. Uracil. Okay, that's in RNA. Okay? If we're talking about DNA, then it would be thymine. Remember, adenine can pair with this, and it can also pair with this. Adenine is a 2-for-1. Right? Okay, next, we also need to know the structures of this. Second thing also, cytosine pairs with guanine. I think we already know that. If you don't, here's a little reminder. Alright, now you know that also. So you know that these pair with these, and these pair with these. Now, we need to know the structure of them. If we actually look at the diagram here, we see that thymine is like 1 pentagon, while the adenine is like 2 pentagons. And you can also see the exact same thing here and here. Now, why is that? Now, we need to know that these are pyrimidines and purines, okay? It's a weird name, but you need to know it, okay? It's called pyrimidines and purines. Now, I'm going to write them down for you. Pyrimidines, which are thymine and guanine, and then there are, my bad, that's the one. And then there are purines, which are adenine and guanine. Now, what does this mean? Oh, wait, and this is, my bad. It's not thymine and guanine, it's thymine and cytosine. I was looking at the wrong thing. Okay, now what does this mean? Now, I just want you to know that the structures of these two are different. Even though that thymine and adenine pair and cytosine and guanine pair, their structures are different. Thymine and cytosine are a single ringed structure. If you remember in Miss Suarez's class, she talked about how carbohydrates almost have a ring-like structure, and that's also another way to identify nitrogenous bases. They also have ring-like structures. But this one, thymine and cytosine, it's just a singular ring-like structure. If you look at the diagram here, we can see how it's like thymine only has 1 hexagon and cytosine only has 1 hexagon, because they only have 1 sugar. While adenine and guanine looks like they have 2, and their structure looks similar to something like this. A hexagon with a smaller pentagon coming out from it. And that makes it a prunine. A little tip to understand is that pyrimidines have a longer name, but the longer name equates to only just 1 ring. And pyrimidines also have now, I don't know how you can memorize, it's because of the Y. Pyrimidines have a Y in it, and because of that, you can know that pyrimidines are thymine and cytosine. Now, prunines, they have a shorter name, but they're the one with 2 rings, right? And to memorize that, you just need to know that if it ends in I, it has I-N-E or pure, you can say adenine and guanine. You can already figure out from the Y that it's going to be thymine and cytosine, and from there you know what the purines are. Now, this is critical to understand because their structures are also the reason why this DNA has a double helix. Now, if we look into this, to put it in perspective for you guys, think of the molecules on one side to another side. I'm going to act as if I split this entire thing in half. So I'm splitting this entire DNA in half. Why am I doing that? Now, if we look at the actual one side here, let's say we take this side, for example, only this one. We're just looking at this here. I'm going to zoom in. Let's see which side has more molecules in them. Remember that the phosphate and the sugar are found in both sides, right? So that means that those are going to be found in both of them. Okay, so those are equating the same amount. Now what we're looking at is the nucleotide, the base pairings itself. This thymine has less molecules than the adenine. Why? Because this adenine has an extra circle here, which means it has more extra atoms. This causes this side of the DNA to have more atoms. So this has more atoms. And why is that important? It's almost like weight is being shifted onto that side. What this causes is almost like a curve, like a slight movement or a bend to that side. Now this side has more atoms here, and it causes shift movement to go this way. And what this causes is almost a double helix shape. So because of the structure of these bases, it causes a double helix shape. Okay, so looking at it, in hydrogen bonds. I needed to talk to you about that. So the reason why there's hydrogen bonding between the two bases is because it needs a sturdy base, but not a strong base. Why is that? Now, when we get to DNA replication, we have to rip the strands apart so that we can make more DNA. And for that to happen, we need something that can easily break apart half of the DNA. And that's why hydrogen bonds are there, because they're sturdy, but they're not strong, right? So you're able to easily break them apart. And the reason why these areas, like the sugar and phosphate, are made out of covalent bonds is because you don't want those to rip apart. You want them to stay connected, or else that DNA is done. It's killed. Okay? Now, any other topic that we have? No. Oh yeah, this is because what happened here was my mic stopped working, so it kind of ruined it. But looking at it, this is everything we need to know. Anti-parallel structures, 3 to 5, 5 to 3, DNA shape, this port here, this here, remember uracil and all that stuff, cytosine, guanine, pyrimidines, thymine, cytosines, and you also know the structures. This is basically everything that's going to be on the test itself. There's literally nothing else. Yeah, that's basically it. Thank you so much. If you have any questions, by all means, text me, email me, shoot me a question. I'll definitely answer it. I hope this helps somewhat.